Hello, and welcome to chapter 3 as we explore life together. So, in this chapter, we discuss the molecules of life. If you recall, in chapter 1, we had the introduction to biology. In chapter 2, we emphasized the fact that living organisms are chemical machines that are made from simple elements that are bonded together, molecules, the molecules of life. So, and then we emphasize the concept of emergent properties, as you add atoms together to form molecules, new properties emerge as you go high in the biological hierarchy. In this chapter, we look at the molecules of cells. So there are over 90 naturally occurring elements, and with man-made elements, they actually are 100. So in this chapter, our focus is on those molecules that are important to life. Remember, we discussed in the previous chapter 25 elements found in humans, in animals, about 17 different elements found in plants needed to support life, building the body, the structural parts of the body, as well as the functional parts of living organisms. So most of the world's population can digest milk-based food. So milk has the carbohydrate lactose in it. And so we will need the enzyme lactase to digest lactose. There are people, however, that are lactose intolerant. They do not have the ability to produce lactase. And so when they eat the food, it stays in their digestive system. It's difficult to digest it. And so they are lactose intolerant. The digestion of milk in human body is used to illustrate the importance of biological molecules. So there are biological molecules, proteins, enzymes in our bodies that helps the chemical reactions, that helps the body metabolisms to be completed, and thereby enabling the daily functions that support the life of the organism to take place. So she's drinking lactose. If you're not lactose intolerant, you're happy. But if you're lactose intolerant, problems follow. Oh my goodness, pain, ache from the indigestion, bloating, and all of those go on. So in this chapter, we have the big ideas that we will discuss. The big ideas: introduction to organic chemistry. Much of the structural and functional molecules of cells are organic compounds. Do not confuse the use of organic compounds in this situation with a common day store organic compound, or the food is organic and the food is not organic. So the organic compounds, and then we're going to discuss the classes of organic compounds, which include carbohydrates, lipids, proteins, and nucleic acids, all of these that are found in the bodies of living organisms that are also produced by living organisms. So the element carbon is very, very important in the diversity of life. So the life's molecular diversity is based on the properties of carbon. Carbon atoms form the backbone of the diverse organic compounds that we know. Carbon is able to do this because of its unique ability to form 4 covalent bonds. Remember carbon, element number 6, the first 2. So element number 6 has 6 protons, 6 neutrons in carbon-12, and 6 electrons. So the 6 of the 6 electrons are distributed, 2 of them in the lowest energy shell and 4 on its outermost shell, which is second electron shell. So with 4 electrons outside, it needs 8. It can covalently bond 1 electron, 1, 2, 3, 4, 4 electrons with hydrogen or some atom of other elements. Because it's able to do this, this is the only element whose atom can covalently bond 4 times with 4 different atoms. And so it's uniquely positioned to be able to form the backbone of this diverse organic compound. So carbon ability to bond with 4 other atoms is the basis of building large and diverse organic molecules. Carbon chains form the backbone of most organic molecules. So the carbon atom in the center can bond to 4 hydrogen atoms, as in methane that we looked at. But then carbon can also bond to carbon, and then the other 3 remaining atoms of each of the carbon atom again bond to hydrogen, as in ethane. So organic compounds can form straight chains, or they can be branched. When they are branched, we call them isomers. So the isomers have the same molecular formula for different structures. Why is it important? Why are we discussing isomers? Because if the straight chain of organic compounds just form long straight chain and cannot bend and change their structure, then we cannot have the kind of 3-dimensional structures that we have in living organisms. Otherwise, we would have miles and miles and miles of strange, long, spider-like organisms. But carbon, being able to isomerize, can take different shapes, different functions. The molecules of cells can take different shapes and different in different cells. So that is very, very important in the structural building of organisms. When carbon is bonded only to hydrogen, we call it hydrocarbons. So it means just a complex containing hydrogen and carbon. So hydrocarbons are composed of only carbon and hydrogen. What are examples of hydrocarbons? Hydrocarbons: methane, ethane, propane, butane, and so on, hexane, and so on and so forth. Hydrocarbons that have all of the carbon electrons covalently bonded to hydrogen are alkanes. Sometimes there's not enough hydrogen, and so the carbon can share 2 electrons and form double bonds, and we would have hydrocarbons that are called alkenes. What are examples of isomers? Let's look at the structural illustrations. So to see this, so so as the isomers are, the molecular formula remains the same and the structures are rearranged. The different arrangement in structures confer different properties to the molecules. And so these have implications in biology. So checkpoint question here. Methamphetamine occurs as 2 isomers. In one form, it is an addictive illegal drug known as crank. And in the other form, it's used as sinus medication. So how could you explain these different effects? Think in biology, structure always follows function. So by changing the function of the organism, you change the function. So remember your checkpoint questions, answered, listed, and emailed to me. Thank you. So this is methane. This is a carbon in the center. This is just a model illustrating. So we don't see the electronic shells, but we know that the outer shell for carbon has 4 atoms. So 1 has 4 electrons, I'm sorry. So 1 electron shared with hydrogen, with hydrogen, with hydrogen, and with hydrogen here. You can see the illustration here in this prism illustration of the model. So this is methane, carbon being able to form 4 covalent bonds by sharing each of its outer 4 electrons with hydrogen. This is a basic hydrocarbon. So this slide shows you examples of hydrocarbons. Remember, hydrocarbons form when carbon bonds to hydrogen only. So easy. So in the previous page, we look at methane. Methane only has 1 carbon. In ethane, carbon bonds to another carbon. So these 2 carbon atoms share their electrons. So there are 2 electrons here. Each carbon now covalently bonds with 3 hydrogen atoms, as in ethane, C2H6. Propane, 1, 2, 3. 3 carbon atoms share electrons. This shares 1 electron with the middle carbon atoms, and the right side carbon atom shares 1 electron with the carbon atoms. Because the central carbon atom is sharing electrons with 2 carbon atoms, it has only 2 electrons left to share with 2 hydrogen. The other 2 terminal carbon atoms are sharing their remaining 3 electrons with hydrogen. So this hydrocarbon is called propane. And with 4 carbon atoms in the carbon chain, we have butane. As the carbon chain lengthens, the isomers can form. So the butane here branches, and you can see it doesn't take as much in linear space. And we distinguish between the normal straight hydrocarbon and its isomer by using isobutane. The prefix isobutane lets you know that it is butane, the same molecular formula, but it's an isomer. So isomers branch. Carbon skeletons may be branched or unbranched. The isobutane is branched. This is normal butane. When the carbon atom is not saturated with hydrogen, they can share more than a pair of electrons. They can share 2 pairs of electrons. They can even share 3 pairs of electrons. When carbon atoms share 2 pairs of electrons, which are 4 electrons, then a double bond is formed, as in butane. 1 butane, because the double bond starts from carbon number 1. The position of the double bond can be started from carbon number 2, and then we have 2 butane. Butane, they are the same, but their double bonds are located originate from different carbon atoms, and so we name them as such. If a carbon atom, not shown in this picture, if 2 carbon atoms share 3 pairs of electrons, then it will be a triple bond. There will be no hydrogen here and probably no hydrogen here, and it will be a triple bond. So the hydrocarbon can be straight chained, it can be branched, it can have double bonds. They can also be form rings in cyclic molecules. So we have cyclohexane and benzene are examples of your ring-shaped hydrocarbons. So this is cyclohexane. So we kind of abbreviate the ring structures by representing the corners, by using the corners to represent carbon. So when you see a ring structure drawn like this means 1, carbon, one carbon, one carbon, one carbon, one carbon, one carbon, 1, 2, 3, 4, 5, 6. So cyclohexane is an alkane. So it's carbon and hydrogen atoms by covalently bonding and sharing electrons are all fully saturated, and there are no double bonds in here. In benzene, on the other hand, which is an alkene, there is not enough hydrogen bonded to every carbon. So the carbon atoms are sharing 2 pairs of electrons in 3 locations. So it's an alkene. And so we have double bonds within the ring. So in this carbon skeleton, so we have carbon, 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 still 6 carbon atoms, but only 6 hydrogen atoms. And so the carbon atoms have to share 2 pairs of electrons, and we have the double bonds in these 3 locations. Cyclohexane has 12. That's all of the electrons that it needs, because it's sharing 2 electrons with this carbon on this end and 2 electrons with this carbon. So it has, this carbon atom has 1, 2, 3, 4 bonds. This carbon atom has 1, 2, 3, 4. Remember, they have 4 electrons on the outermost shell. This carbon 1, 2, 3, 4. This carbon atom 1, 2, 3, 4. In benzene, however, this carbon atom has 1, 2, 3, 4, because 2 of the bonds are formed by the sharing of 2 pairs of electrons between 2 carbon atoms. So isomers are the same, they're molecules that have the same
Isomers are compounds that have the same molecular formula, but different 3-dimensional structures. The difference between isomers can literally mean the difference between life and death. L-DOPA, shown on the left, reduces the symptoms of Parkinson's disease. Its isomer R-DOPA on the right has no effect on the disease.
So these are enantiomers. They are mirror images of one another. If you turn this flip order, these 2 are mirror images of each other, L-DOPA and left-sided DOPA and right-sided DOPA. So if you turn the molecule left, you get L-DOPA. You turn the compound left. If you turn the compound right, then you have the R-DOPA. But even that structural change means a significant difference in fusion, which impacts life differently.
The carbon backbones of organic molecules may vary in length and in the location of single and double bonds. Molecules may be straight or branched and may even contain rings of atoms. Three types of isomers are structural isomers, which differ in the covalent partnerships between their atoms, geometric isomers, which vary in arrangement of atoms around a double bond, enantiomers, or stereoisomers, which are molecules that are mirror images of each other, like left and right hands.
So there are a few groups of compounds that are key to the functioning of biological molecules. So very important. So we are focusing on organic compounds because they're very, very important for the functioning of biological molecules. The properties of organic compounds depend on the size and shape of its carbon backbone and the atoms that are attached to that skeleton. So size, shape of the carbon backbone and the atoms that are attached to that molecule. Some molecules have hydrophilic functional groups. Hydrophilic, water, other functional groups. So Hydrophilic have affinity, hug water. So hydrophilic functional groups give organic molecules specific chemical properties. And let's look at the table 3.2 to look at the important chemical groups. So hydroxyl group is found in, for example, alcohol is the OH group. Definitely has affinity for water. So it's hydrophilic, it's water loving. So it would mix and it would dissolve into solution in water. The carbonyl group COO, if it is in a carbon atom all by itself, then it attaches to the hydrocarbon and forms a ketone. If it attaches to a carbon atom that binds to another hydrogen, it forms aldehyde, so it forms aldehyde. So carbonyl groups bonded to the hydrocarbon chain results in the formation of ketone or aldehyde. The carboxylic group bonded to the hydrocarbon chain makes the organic acid, the carboxylic acid, like citric acid and so on and so forth. Dissolves in water and it ionizes to give hydrogen ions in solution, so it's an organic acid. The amino group, NH2, when it binds to the hydrocarbon, makes it an amine. So these are very important. In amino acids, we have these two functional groups. This is the ionized form of an amino group. Phosphate group binds to an hydrocarbon. That's how we have the high energy compounds of adenosine triphosphate. And methyl group, methylation of the compound, all of these joining to the hydrocarbon affects the function. So hydrocarbons, organic compounds that have the carbon backbone, they are very important biological molecules. The properties of these biological molecules is determined by the size and shape of its carbon skeleton, as well as the atoms that are attached to it. The atoms that are attached to the carbon skeletons are called functional groups. In biological systems, because of our water origins and the composition of the water, the functional groups tend to be hydrophilic. They are water loving. The body is 70% water, and so it's essential that the functional groups to be able to interact with them be hydrophilic. There are 6 important functional groups. They include the hydroxyl group. When it binds to the hydrocarbon, it forms alcohol. There's a carbonyl group. When it binds to the hydrocarbon, it becomes a ketone or aldehyde. There's a carboxylic group. When it binds to the hydrocarbon chain, forms an organic acid, a carboxylic acid. And like all other acids, it can dissociate in solution to give hydrogen ions. The amino group combines with the hydrocarbon to give an amine. These two are important components of proteins, the amino acids that work in the body. Phosphate group binds to hydrocarbons to give organic phosphate, the high energy molecules that our bodies use. Methyl group also to methylate the compound. So checkpoint question, remember to answer them. Identify the chemical groups that do not contain carbon. Of these 6, which chemical group does not contain carbon? And email them to me. So a few chemical groups are key to the functioning of biological molecules. As we saw in an image earlier, the sex hormones, testosterone and estradiol, they are, this is a type of estrogen. So male, female hormone. They differ in the groups of atoms that are shown here. So this is testosterone, this is estradiol. These are ring molecules. They are steroids, so they have the characteristic 4-member ring, but they only differ from one another on their side chains. So there's O here in testosterone, OH in estradiol, and then the methylation, instead of being on the first ring, is on the third, is missing in estradiol. And look at this, testosterone producing male and the main, so different from the female, just on the basis of differences in these side functional groups. So the chemicals, the structure of the chemicals that make up the body parts eventually results in the magnified appearance that we see in the organisms that they constitute. Cells make large molecules from a limited set of small molecules. So there are 4 classes of biological molecules that contain very large molecules. So the molecules that are large in size, we call them macromolecules. Macro means large, so macromolecules have large sizes, so large molecules. Another name for macromolecules are polymers, because there's not a single element that's just large by itself, but several atoms of different elements combine to form molecules, and the more elements, the more atoms that constitute that molecule, the larger that molecule will be. So that makes sense, right? So they're called polymers because they are made from identical or similar building blocks that are strung together. Just as in the building of a house, the blocks or the pieces of panels are put together, or just like you put your pieces of Lego together, you can build a castle from relatively small pieces. So the building blocks for polymers are called monomers. So the 4 classes of biological molecules contain large molecules. We call them macromolecules. Those macromolecules are also called polymers because they are built just like a house is built from small bricks that are put together to build make a big building. The monomers, the smaller molecules that build the polymers, are called monomers. So how do monomers help to build polymers? They do so by what we call dehydration reactions, and we're going to look at examples of those, illustrations of those in the next slides. Polymers can be built up by dehydration reaction, in which 2 monomers come together, and through their reaction, they give up water. Because water is removed, one molecule gives hydrogen, the other molecule gives hydroxyl group, and water is removed. It's called dehydration reaction. Polymers that are built that way can also be broken down by hydrolysis. So when plants make photosynthesis, they make glucose, and many glucose molecules are joined together in the plants to form the potato tubers, the corn, the carrots. Either the monomers, when two monomers join together, they form dimers. Many monomers together form polymers. So the formation of dimers and polymers involves the removal of one molecule of water, and so it's called dehydration reaction. When you and I eat the food from the plants, the corn, and then we need to break down the starch or the disaccharides, then we add the water back in a reaction called hydrolysis. So these reactions are mediated by enzymes, but they're always taking place in the bodies of living organisms. So checkpoint question, remember to compile them and email them to me. Suppose you eat some cheese. What reactions must occur for the protein of the cheese to be broken down into its amino acid monomers, and then for these monomers to be converted to proteins in your body? So remember, answer the question and email it. So we said that the chemical molecules in our bodies are macromolecules that are made from monomers. So in this illustration, each purple ball is like a monomer, a building block. So it reacts with an, it bonds. Each of these is a monomer. Each of these purple is your carbon atom. The OH is a functional group, and so this is carbon, carbon, carbon. Now, this is one monomer, this is another monomer. These two monomers are going to join together. When they do so, the 3-carbon monomer is going to give its OH. The 1 carbon monomer is going to give its H. These two combine and release water. This is where the name dehydration reaction comes from. Now, this that was previously bonded to hydrogen and this polymer that was previously bonded to OH will no longer bond to OH, no longer to H, but now they bond to one another. And so we go from 3 carbon atoms to 4 carbon atoms. This is a longer polymer. The process can repeat and repeat. You can have a very long chain of polymer forming in there. And when that polymer, gets into another organism, it can be broken down simply by adding the water, and the water goes back to its original position, and we have the original compounds there. Okay.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
Now let's take the different important classes of biological molecules. So we look at carbohydrates first. As the name suggests, they are made from carbon, hydrogen, and oxygen, usually in the ratio of 1 carbon to H2O. So CnH2O. So the simplest form of carbohydrates are monosaccharides. So they're just one unit of sugar. So they're all monomers. The carbohydrates, they have a different number of carbon atoms, but they're all one molecule. So carbohydrates range from those simple sugars which are made of just one monomer to large polysaccharides with many monomers joined together. So the sugar monomers are called monosaccharides. The general formula for monosaccharide is CH2O, and it contains the hydroxyl group and the carbonyl group. So hydroxyl group and a carbonyl group. So checkpoint question is write the formula for a monosaccharide that has three carbon atoms. So this is honey, sweet, and a carbohydrate, and meat on there. So it's this is the example of another carbohydrate or monosaccharide. I'm sorry, is glucose. Glucose and fructose are isomers. And so glucose as its carbonyl group is terminal, and carbonyl group on fructose is on carbon number 2, is in the middle of the chain. And this guy, fructose, is the sweetest member of the monosaccharides. Of course, we use monosaccharides as sweeteners in food, and insects, other animals love their sweet taste as well. We number the carbon atoms 1, 2, 3, 4, 5, 6. They can remain in straight chain, but in biological systems, they rarely exist. They usually form the chair or the boat from which the rings kind of close. But these are isomers, glucose and fructose. So this is the structural formula. So the first top right hand corner is the carbonyl group stays oxygen, and we have the carbon number 1, 2, 3, 4, 5. Why are we looking at this? Remember, it said this fructose and glucose in biological system in natures, they are rarely found in this straight chain format. Rather, they exist in the ring form. So this is the structural formula that shows how the structure actually is in the cell. So the corners, except for the top right hand corner, are represented by the carbon atoms. Okay, the carbon atom with the functional group on the outside. To make it easier and abbreviate, in this abbreviated structure, notice that we no longer have the carbons written. The corners simply represent the carbon atoms. The only corner that's the carbon atom retains its title so that we know that oxygen is there. And in the simplified structure, which you see in a lot of textbooks, only the oxygen corner is recognized, and the rest of the structure by familiarity is identified and recognized as such. When two monosaccharides are linked together by dehydration reaction and they now form a covalent bond in between, then they become a disaccharide. Di means two. So if three monomers bond together, they will form a trisaccharide. Beyond that, actually these days it's from 3 and above, we just call them polysaccharides. So the checkpoint question, lactose, as you read in the chapter 1, is the disaccharide sugar in milk. It is formed from glucose and galactose. The formula for both these monosaccharides, it's C6H12O6. So glucose, galactose, fructose, they all have the formula C6H12O6. but they have a different shape. What is the formula for lactose? Again, answer your checkpoint questions and email to me. So when one glucose molecule bonds with another glucose molecule by dehydration reaction, one glucose molecule will give OH and the other gives H. That becomes water in a dehydration synthesis. So the dehydration goes away. The two molecules of glucose join together and they form the disaccharide maltose. So glucose plus glucose forms maltose.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugar cane and sugar beet and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of two linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
So 3 examples of disaccharides: sucrose, lactose, maltose. Remember their compositions. Are we eating too much sugar? I will send you a video to watch on sugar and the impact on us. The FDA recommends that only 10% of the daily calories will come from added sugar, but we are doing way too much than that. And research supports the correlation between high sugar intake and adverse health effects. And so some of the people that are suffering even the comorbidities from this pandemic are complicated from their past experiences of sugar and conditions such as diabetic CDC. Sugars are often described as empty calories. What do you think that means from a nutritional standpoint? Remember your checkpoint questions and email. So this is our description of sugar consumption. This is World Health Organization's recommendation that annually we consume 20 pounds of sugar. This is the FDA, the US FDA recommendation, doubling that 40 pounds. But this is what the average American eats in sugar, 130 pounds. And it's all over the world. People are consuming and gobbling sugar up, and it's doing a lot of damage in our bodies. So polysaccharides are long chains of sugar units. Examples of polysaccharides are starch and glycogen. So starch is the storage polysaccharide in plants. Glycogen is the storage polysaccharide in animals. Other polysaccharides include cellulose. This is the most common, most abundant polysaccharide on Earth. It is the material that forms the cell walls of plants. Chitin is a component of insect exoskeleton and is in the fungal cell wall. So the mushroom lovers, you get chitin. It's a very, very high and good fiber, and it locks in vitamin D's and B vitamins. So checkpoint questions, remember to answer them and email. Compare and contrast starch and cellulose. These are two plant polysaccharides. So these are in your potatoes, the starch granules in the cells. So this is an electron micrograph looking inside the cell. So in addition to the organelles, these are granules that are stored in the potato tuber. I will send you some video to watch. We normally will observe this in the laboratory, in the real laboratory under the microscope. We're not able to do that because of the pandemic, but I will email you videos to watch. And So they are the starch granules are built from molecules of glucose that are used to form polymers of starch. In the animals, which is in the muscles of this lady, we observe that under the microscope. So we see the glycogen granules in the muscles. So look at the branches, the fibroid manner in which they are put together is still to form glycogen. So we have the same glucose molecules, but the way in which they are arranged determines whether they will become starch or glycogen. In plants, the same glucose molecules are put together to form cellulose microfibrils in the plant cell walls. And you can see one fiber, two, three, four, and each fiber is a long stretch of a polymer of glucose. And then the hydrogen bond with one another and wound around one another to form the fibers of the cellulose. So the structural arrangement of the molecules very, very important in function.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Now we look at the next class of organic compounds, the lipids. This includes fats, lard, and all the oily substances. They are diverse in nature, but they have the common property that they are hydrophobic. They have a phobia for water. They do not love water. So lipids are diverse hydrophobic compounds. They're composed of largely carbon and hydrogen. Examples include fats. Fats are triglycerides. We call them that because they consist of glycerol. Glycerol has 3 carbon atoms, each with hydroxyl group. In fatty acids, the hydroxyl groups are replaced with organic acids, with the carboxylic acid. Some fatty acids have double bonds in them and they are referred to as unsaturated fatty acids. Unsaturated fatty acids are common in plant oils. They tend to be fluid at room temperature and tend to be a part of a healthy diet. Fats that have maximum number of hydrogens are called saturated fatty acids. Like in animal fat, they tend to be solid at room temperature. So, because of our knowledge of saturated fats, commercial industry started hydrogenating vegetable oils, plant oils, so that they begin to be saturated and they are converted to saturated fat. So margarine, I don't believe it's not butter on all of those products. They are actually vegetable oils that have been saturated with hydrogen so that they solidify and they can become spreads that we can eat. This hydrogenation, however, when you use them, creates trans fats, which are associated with health risks. So again, I will email you a video to watch on trans fat and our health. So explain why fats are hydrophobic. Remember, your checkpoint questions should be emailed to me. So this is glycerol. Remember, I described it as 3 carbon atoms and 3 functional hydroxyl groups attached to each carbon atom. The rest of it is hydrocarbon. To become a lipid, the glycerol, through dehydration synthesis, binds with a fatty acid and it becomes, water is removed and we have a monoglyceride. So the monoglyceride is a fatty acid. When the second OH group forms a bond with another fatty acid, then we have a diglyceride. When the 3 hydroxyl groups form a bond with 3 fatty acids. When the glycerol is bonded to 3 fatty acids, then we have a triglycerides. So the fats that we are familiar with, that we use in our kitchens and consume daily, are triglycerides. They are glycerol that are bonded to 3 fatty acids, triglyceride. So this butter and the fat in your beef are saturated fats. In olive oil and salmon, we have unsaturated fats.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long, nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in 1 or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats, like butter and lard, are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
So, by the 1990s, partially hydrogenated oil were common in countless foods. I mentioned a few, and I will send you the video of scientific studies that document the health risks of trans fats. So they pose greater risk even than saturated fats. So in the scientific studies, there's an experimentally controlled feeding trial where diets contain different proportions of saturated, unsaturated, and partially hydrogenated fats. And the outcome of the result was that the trans fats pose even greater risk. So, checkpoint question. What is the difference between retrospective and prospective study? I think that's a repeat question. Go ahead and answer it again and send it to me with your email for today's class. So this chart illustrates the increased risk if we have diets that's high on polyunsaturated. We have decreased 75% decrease in risk, 50% decrease in risk, all of this green band. But when we go to monounsaturated fat, we have the baseline, no risk different. As we increase saturated fat, trans fat, we progressively increase. As we increase the amount of trans fat, we progressively increase our risk of heart disease and health. So, okay, another group of lipids are called phospholipids and steroids, and these are important lipids, and they have a variety of functions in living organisms. Phospholipids are very important. They make up the cell membranes of all living organisms. The biological membranes are made of phospholipids. Steroids include cholesterol and some of the hormones. We looked at testosterone, estradiol, and so on and so forth. And then cholesterol is found in animal cell membranes, very important composition. It's also the precursor for making many of the steroids, including testosterone, estrogen, the sex hormones that we have. So checkpoint question, remember to answer that. Compare the structure of the phospholipid with that of the fat. So we should say a phospholipid has a fat side, which is the glycerol, the fatty acid tails, but in place of instead of being a triglyceride, a phosphate group will combine to the other. Now, remember, these regions are hydrophobic. With the addition of the phosphate group, we bring an hydrophilic head. So this is a phospholipid. It's a diglyceride with a phosphate group on it. And because the phosphate group is hydrophilic and the hydrophobic tails are hydrophobic in an aquatic environment, they spontaneously hydrophobic heads move towards water and hydrophilic heads shy away from water, resulting in a double layer of what is called the phospholipid bilayer. And that's how the biological membranes are constructed in living organisms. This is a steroid showing here. All of the steroids would have this 4-member ring, and they will be one different from another by their side chains. So there will be a discussion point going out on anabolic steroids today for our current thoughts. These are synthetic variants of the male hormone testosterone that athletes abuse in order to advance their careers. So we're going to discuss that on the discussion board. Checkpoint question: explain why fats and steroids, which are structurally very different, are both classes of lipids. So steroid helps you build all of this, gain undue advantage, but they also do a lot of damage in the body. They not only cause the muscles that you want to run with to enlarge, but muscles of internal organs, which can result in fatality. Proteins, another class of organic molecules, very, very important. So proteins are very, very important. They are involved in nearly every dynamic function in the body. And there are several types of dynamic functions in the bodies of living organisms. So let's list the functions of proteins. So proteins, important structural and functional parts of living organisms. Now we're looking at the functional side. They function as enzymes. All of those chemical reactions that take place in our bodies to make the body function, many of them will take forever to complete without the help of enzymes. But the enzymes help this speed up the rate of chemical reaction, helping the body to be able to respond to changes in the environment and bodily needs on time to sustain life. Proteins function as transport proteins that are embedded in the cell membranes. So certain materials that will be destructive and cannot go through the membrane directly, they would have to be carried by proteins in and out of cells. There are defense proteins in the body that make, such as antibodies. Right now that we are going through the COVID-19 pandemic, we are looking at antibodies that defense proteins in the bodies are made to see if that could be used to prevent infection and or severity of disease in others. Signal proteins such as that, as many hormones, help to relay signals, information from one part of the body to another, telling the body, oh, you just had a baby, it's time to start making milk to feed the baby. Such important functions. There are receptor proteins that recognize foreign bodies, antigens, and let your body know, hey, you got a bacterial infection, and so on and so forth. There are contractile proteins found within muscle cells. In the case of humans, there are also contractile proteins found in small organisms, even protists, that helps them to be able to move around in the waters that they have. Actin, myosin. The structural proteins such as collagen, you're all trying to buy collagen to look young, to make your skins far much stronger and more flexible, and storage proteins that are in the bodies of living organisms. So proteins have its wide range of function and structure. Proteins are composed of different arrangements of a common set of just 20 amino acids. So the 20 amino acids, like the letters of the alphabet, can be put together to form different structures that perform different functions in the human body. So the functions of different types of proteins depend on their individual shapes and their individual composition. The individual shapes depend on the individual amino acids that are put together, and their original identity determine their interactions with one another. Proteins can be denatured, and in the process of denaturation, a protein unravels and loses its specific shape. When it loses its specific shape, it loses its function. In your kitchen on a regular basis, you denature proteins. You take an egg, you fry it, it loses its jelly-like appearance, and it becomes more solid and rigid. If you were to try to work with the enzymes, it no longer work. Why does a denatured protein no longer function normally? When we denature the protein, we break the bonds and we rearrange, distort the molecules, and they can no longer function. So remember to answer your checkpoint questions and email to me. So proteins have different levels of organization, and their function must be their shape. So look at this protein molecule, for instance, has grooves here where the target molecules bind. If you raise the temperature or pour an acid and change that shape, the group will no longer be there to fit the molecules that normally bind, and the enzyme will not function. So this is illustrating the spider putting proteins together to form these webs in a neat arrangement. So the building block for proteins are amino acids, and when two amino acids bind together by dehydration reaction synthesis, they form peptide bonds. Two proteins form a dipeptide, three form a tripeptide, and then we have polypeptides. Protein diversity is based on the different sequences of the amino acids that are the monomers that they contain. So what is an amino acid? An amino acid is a monomer that has an amino group, a carboxyl group, a hydrogen atom, and an R group. That R group, all of these 1, 2, 3, 4 are attached to a central carbon atom, but the R groups distinguish 20 amino acids, each with specific properties. So this is the description of an amino acid: a central carbon bonded to by hydrogen, carboxylic group, an amino acid. This R group will be varied, and that's what will distinguish one amino acid from another. Amino acids are unique. They're the only ones that have two functional groups. The amino group is a functional group. Carboxylic acid is another functional group. This R could be just CH3, or it could be CH3, CH, then it could be long chains. And all of the amino acids will have this purple, gray area in them, but they will differ from one another by their R groups. So the amino acid monomers are linked together in a dehydration reaction, joining the carboxylic group of one amino acid to the amino group of the next. And the bond that they create is called a peptide bond. And when several amino acids are bonded together, a polypeptide is formed. So remember your checkpoint question: by what process do you digest the proteins you eat into the individual amino acids? if you build them up by dehydration synthesis, what would be the way to digest them? So some amino acids are hydrophobic. They don't love water. Other amino acids are hydrophilic, water loving. So this here does not have a water loving side. This has the OH group which loves water, and the COOH which loves water. So leucine is hydrophobic. Serine and aspartic acid are amino acids that are hydrophilic. So you can see how the R groups influence the properties of the different amino acids. So this is showing the dehydration synthesis of proteins from. So this is one amino acid, the second amino acid, and the dehydration reaction takes place. A bond is formed now between carbon and nitrogen. The carboxylic acid group of one amino acid gives the OH group away, and the amino acid of the second amino acid gives the H away, forming water in the dehydration reaction. Now a new covalent bond called the peptide bond is formed between carbon and nitrogen, and the product is a dipeptide. Di meaning two. It has two amino acids bonded together by peptide bond in a dipeptide. So a protein can have 4 levels of structure. The primary structure is simply the sequence of amino acids joined together by dehydration reaction. You have long chain or polypeptide chain. Secondary structure is formed by the coiling and folding of the chain as the members of the chain hydrogen bond with one another, and the hydrogen bond stabilizes them. In the tertiary structure, we have the 3-dimensional shape because of the interaction between the R groups. So they can form disulfide bonds, more hydrogen bonds, and they simply wind the chain tighter together. And the quaternary structure, proteins are made from one or more polypeptide chain that come together in the quaternary structure. So remember your checkpoint question. If a genetic mutation changes the primary structure of the protein, how might it destroy the protein's function? So this is still the primary structure of protein. Peptide bond 1, peptide bond 2. In that way, glycine bonded to proline, to triosine, to glycine, triosine, glycine, and so on and so forth. And you have all the cysteine, proline, leucine, valine, lysine, and you have all this long chain of polypeptide primary structure. Now, in the secondary structure, you have hydrogen bonds, second hydrogen bonds forming between atoms of the backbone. So hydrogen bonds are forming between atoms of the backbone. As a result of the hydrogen bonds, 2 types of secondary structures can form: the alpha helix, like the spiral spring, or the beta plated sheets that you have in here. Then the R groups in the tertiary structure, the R groups interact with one another, winding the molecules into more globular shape. And then the quaternary structure, different tertiary structures can come together. In this case, it's 1, 2, 3, 4 coming together to make a functional protein.
Most proteins are folded into a complex globular shape. Each protein molecule consists of 1 or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure. Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugation. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where 2 regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall 3-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of 2 or more polypeptide chains. The 4th level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
So finally, we look at nucleic acids. Nucleic acids are called nucleic acids as they were first isolated from the nucleus of the cell. Examples of nucleic acids include DNA and RNA. These are information-rich polymers. The monomers for making the nucleic acid are called nucleotides. Each nucleotide consists of a sugar, phosphate group, and nitrogenous base. 3 parts of nucleotide: sugar, phosphate, and nitrogenous base. The DNA molecule, deoxyribonucleic acid, is a double helix. The RNA molecule, ribonucleic acid, is a single chain of polynucleotide. Working together, DNA and RNA serve as the blueprints for the synthesis of proteins, and they control the life of the cell. The DNA molecule is described as a molecule of inheritance. It's the only self-replicating molecule that we know. Okay. Sorry, excuse the technical. My PowerPoint kind of, I had to force quit and start again. So the monomers that make up nucleic acid are called nucleotides, and DNA is the molecule of inheritance. It is a double helix, and the DNA is deoxyribonucleic acid, RNA is ribonucleic acid. DNA is a double helix, RNA is a single strand of polypeptide chain. So this is a nucleotide, sugar, phosphate, nitrogenous base, in this case adenine. In RNA, we will find 4 adenine, with cytosine, guanine, and uracil. In RNA, we'll find 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. So in the DNA or RNA, the sugar and phosphate part of the nucleotide will form the backbone of the chain, and the nitrogenous bases will stick out on the side, and so they can interact with other nucleotides and so on. So this is adenine, thymine, cytosine, guanine. And the DNA molecule, there's double strand as the nitrogenous bases form bonds, hydrogen bonds with one another. Thymine double bonding with adenine, cytosine triple bonding with guanine. And this is the DNA. So this is a base pair. It's a base pair rule in which adenine would always bind to thymine, cytosine would always bind to guanine. Repository of information, the control center of the cell. On the DNA, long strand of DNA are specific genes as previously discussed, and the information, which is in the form of a sequence of nucleotide, is transcribed, used as a template to transcribe the RNA molecule. The RNA molecule leaves the nucleus. Remember, they are called nucleic acid because of the DNA typically found in the nucleus of the cell. And so RNA then gets the instructions, the information that's stored in the DNA by transcription, carries it to the rest of the cytoplasm. That information is used to is translated into the making of amino acid polypeptide protein. So very important molecules of life. Checkpoint question: what role does complementary base pairing play in the functioning of DNA? So explain how lactose tolerance involves 3 of the 4 major classes of biological molecules. So it is known that some people are lactose tolerant, others do not. Different mutations in DNA in humans have led to lactose tolerance in several human groups. Both their ancestors raised cattle. Those whose ancestors raised cattle have developed the lactase gene permanently turned on in their body, and so they do not suffer lactose intolerance. So milking, different ways of milking. So you should be able to, at the end of this chapter, describe the importance of carbon to life's molecular diversity, Describe the chemical groups that are important to life. Explain how a cell can make a variety of large molecules from a small set of molecules. Define monosaccharides, disaccharides, and polysaccharides, and explain their functions. Define lipids, phospholipids, and steroids, and explain their functions. Explain how trans fats are formed in food. Describe the evidence that suggests that eating trans fats is more unhealthy than consuming saturated fats. Describe the chemical structure of proteins and the importance of protein to cells. Describe the chemical structure of nucleic acids. Explain how they relate to inheritance. Explain how lactose tolerance has evolved in humans. Thank you.